I want to start today with something personal. There is a particular kind of grief that doesn't get talked about enough in medicine, and it's the grief of watching someone you love lose themselves. Not their body, not their mobility, but them. Their recognition of you, their memory of who you are to each other. Professor Tim Spector, one of the most cited scientists in the world, a man who has spent decades at the frontier of microbiome research, has spoken publicly about watching his mother descend into dementia, about reaching a point where she no longer recognized him, about sitting across from a person who was physically present, but in every other sense, gone. That experience, combined with his own mini-stroke in 2011, sent him on a research journey that fundamentally changed how he, and increasingly how the broader scientific community, thinks about the relationship between the brain and the rest of the body. And what he found, what the last decade of research in this space has produced, is a story that is, to me, one of the most paradigm-shifting things happening in medicine right now. The story, in its simplest form, is this. To protect your brain, you have to stop thinking about the brain as the center of the story. You have to look somewhere else entirely. You have to look at your gut. Today I want to walk you through why, and I promise, by the end of this episode, the phrase "trust your gut" is going to mean something very different to you. For most of the history of modern medicine, there was a very clean, very confident distinction between two domains. On one side, the brain, the seat of consciousness, personality, emotion, cognition, the domain of psychiatry and neurology. Protected behind the blood-brain barrier, a highly selective filter that prevents most substances in the bloodstream from entering the brain's environment. The brain was treated almost like a sovereign organ, separate, elevated, isolated from the messiness of the rest of the body. On the other side, the body, the mechanical system, organs, tissues, metabolism, immunity, all the biological machinery that keeps us running. The domain of medicine, of physiology, of gastroenterology. The brain was the mind, the body was everything else, and you treated them accordingly. This division has shaped medical training, clinical practice, diagnostic categories, and drug development for well over a century. Psychiatrists treated the mind. Gastroenterologists treated the gut. Rarely did these specialties consider that they might be treating the same system. And here's the thing, that separation was always somewhat artificial. The biology was never actually divided the way the institutional categories were. But the tools to see the connection clearly weren't there until relatively recently. The science of the microbiome, the trillions of microorganisms living in and on the human body, was in its infancy just 20 years ago. The gut-brain axis, as a research field, barely existed in mainstream discourse a decade ago. What the last 15 years of research have done is not just add nuance to existing understanding, they have fundamentally restructured the model. Professor Tim Spector, when he began his deep investigation into these questions, articulated something that I think captures the shift beautifully. He talked about increasing lifespans, people are living longer, but declining health spans. The years of life are extending, but the years of good life, of cognitive vitality, of mental clarity and physical function are not keeping pace. There is a widening gap between how long we live and how well we live. And his research kept pointing to the same place as a primary driver of that gap. Not genetics, not primarily, not just lifestyle in the generic sense. The gut, the microbiome, the ecosystem of trillions of microbial organisms that we are hosting, and in many cases, inadvertently starving every single day. The central claim of today's episode, backed by some very serious epidemiological and mechanistic research, is that the gut is not a support system for the brain. In evolutionary terms, in communication terms, in inflammation terms, the gut is running the show. And the brain is, to a far greater degree than we ever acknowledged, listening. Let me show you what that looks like in practice. Starting with something you probably do every morning and may never have thought about as a brain health intervention. When was the last time you flossed? I know, you've heard it from your dentist. You've nodded along. You've probably said, "I'll do better," and then mostly forgotten about it until the next checkup. Most of us have. But I want to reframe this conversation entirely, because the stakes are considerably higher than a cavity. The connection between oral hygiene and cognitive decline is something that has accumulated a striking body of evidence over the last decade, and I think it remains one of the most underappreciated findings in all of preventive medicine. So let me walk you through the mechanism, because once you understand it, the habit becomes very easy to take seriously. Your mouth is not a sterile environment. It is home to hundreds of species of bacteria, living in the soft tissue of the gum line and in the biofilm we call plaque. In a healthy mouth, these bacterial communities are largely benign, kept in balance by regular brushing, flossing, salivary defenses, and mechanical disruption. In a mouth with poor hygiene, where plaque builds up, where gum disease develops, those bacterial communities become dysregulated, and crucially, the gum tissue, when inflamed, becomes permeable. The microbes and the inflammatory compounds they generate start leaking through the gum lining directly into the bloodstream. This is not a dramatic event. It's gradual. It's a slow, low-level, continuous seepage of microbial and inflammatory material into systemic circulation. Hence the phrase I think captures it perfectly: a slow leak of inflammation. Now the brain has that blood-brain barrier I mentioned earlier, that protective filter. But the filter is not impenetrable. Under conditions of sustained systemic inflammation, the blood-brain barrier becomes more permeable than it should be. Microbial compounds, inflammatory markers, lipopolysaccharides from bacterial cell walls. These materials enter the brain environment and they trigger local neuroinflammation, inflammation in the brain itself. And what does chronic neuroinflammation look like over years and decades? It looks like the protein tangles and cellular damage we associate with Alzheimer's disease and other forms of dementia. Specific periodontal bacteria, including one called Porphyromonas gingivalis, have now been found in post-mortem samples from the brains of Alzheimer's patients, in concentrations that correlate with the degree of cognitive impairment. This is not a theoretical pathway. The bacteria are physically present in the brains of people with dementia, and in most cases, they got there through the gum line. The epidemiological data puts a number on this that I find remarkable. People with poor oral hygiene, chronic gum disease, high plaque burden, have approximately double the risk of developing dementia compared to people with good oral hygiene. And conversely, flossing regularly, which disrupts the plaque, removes the bacterial colonies before they can establish the chronic low-level inflammation, has been associated with a nearly 50% reduction in dementia risk in some studies. Half, just from flossing. Now, I want to be careful here to say this is not a simple causal claim. People with good oral hygiene also tend to have other healthy habits. The relationship is complex, but the mechanistic pathway is real and well described. The bacteria are getting into the brain, the inflammation is happening, and the tool to interrupt that pipeline, dental floss, 30 seconds a day, is one of the most cost-effective neuroprotective interventions in existence. The next time you pick up your dental floss, I'd like you to think of it not as an oral hygiene tool, but as a brain health tool, because that's what the biology says it is. I want to come back now to a concept I've discussed in detail in a previous episode, the 80-20 rule of the vagus nerve, but I want to approach it today from a slightly different angle. Because in the context of the gut-brain axis, this asymmetry takes on a meaning that goes beyond just stress physiology. The vagus nerve, as I've described before, is the primary neural highway connecting the gut and the brain. It's a massive, widely branching nerve that runs from the brainstem down through the chest and into the abdomen, carrying bidirectional communication between the central nervous system and the digestive system. And that communication is not symmetric. Approximately 80% of vagal signal traffic flows upward, from gut to brain. Only 20% flows downward, from brain to gut. So let's sit with what that actually means. We've been operating, clinically and culturally, on the assumption that the brain is the controller and the gut is a receiver. But the anatomy of the primary communication channel between the two says the opposite. The gut is sending far more information to the brain than the brain is sending to the gut. The brain is not a commander. The brain is, in a very real functional sense, a listener. A highly sensitive, highly responsive listener to the constant stream of data arriving from below. This has an evolutionary explanation that I find genuinely fascinating. The enteric nervous system, the network of neurons that lines your entire digestive tract, is extraordinarily complex. It contains somewhere between 200 million and 500 million neurons. That's more neurons than in your spinal cord. And here's the thing that really puts it in perspective. In embryonic development, the enteric nervous system forms before the brain. The gut nervous system is, evolutionarily and developmentally, our first brain. The central nervous system, the brain in our skull, came later. It grew on top of and in constant communication with a system that was already there, already running, already managing the extraordinarily complex business of keeping an organism alive. This means that what we experience as brain fog, as sudden dips in mood, as inexplicable anxiety that doesn't seem connected to anything in our environment, these are often not originating in the brain at all. They are the brain receiving and responding to dispatches from below, status reports from the gut, messages from the microbiome. When your gut microbiome is healthy and diverse, the messages it sends upward are calm, regulatory, reassuring. When the microbiome is dysregulated, low in diversity, high in inflammatory species, starved of the fiber and polyphenols it needs, the messages change. They carry inflammatory signals, dysregulated hormonal cues, the chemical language of a system under stress. And the brain, the sensitive listener at the other end of that 80% upward channel, responds accordingly. Brain fog is a gut signal. Unexplained low mood is often a gut signal. The sense of being wired but tired, anxious without clear external cause, is often a gut signal. This reframing changes everything about how we approach these conditions. Instead of asking, "What is wrong with my brain?" the more productive first question is often, "What is my gut sending my brain right now?" I want to be thoughtful here, because the topic I'm about to address involves something that affects hundreds of millions of people worldwide, and I don't want to oversimplify. But the science has moved to a point where I think it would be irresponsible not to discuss what's emerging in this area. For most of the last several decades, the dominant model of depression in mainstream psychiatry has been the chemical imbalance theory. The idea that depression is fundamentally a deficiency of neurotransmitters, primarily serotonin, sometimes dopamine, and that the way to treat it is to restore those levels pharmacologically. This model produced SSRIs and SNRIs, the antidepressant medications that hundreds of millions of people take worldwide. And these medications do work for many people, to varying degrees. I am not dismissing that. But there has been growing recognition in the research literature that the chemical imbalance theory is an incomplete picture. It doesn't explain why a significant proportion of people with depression don't respond to serotonergic medications. It doesn't explain why the same antidepressant works powerfully for one person and not at all for another. It doesn't explain why depression rates are rising in populations with increasing access to treatment. What the emerging evidence increasingly points to is something called the immunobasis theory of mental health. The idea that chronic low-grade systemic inflammation, not a deficiency of serotonin, is the primary driver of many cases of long-term depression. And the entry point for that inflammation, in many cases, the gut. Here's the clearest illustration of this mechanism I can offer. Think about how you feel in the 24 to 48 hours after receiving a vaccine. Most people feel tired, a bit low, withdrawn, not quite themselves. There's a flatness to mood, a disinclination to engage, a kind of mild but real malaise. That is not a coincidence. What you're experiencing is your brain responding to an immune activation signal. The vaccine triggered your immune system to mount a response. And one of the downstream effects of that immune activity is a set of signals that reach the brain and induce what researchers call illness behavior. The reduced activity, social withdrawal, and low mood that evolved as a protective mechanism during genuine infection, when staying still and conserving energy was adaptive. Your brain doesn't know you got a vaccine. It just received immune activation signals and responded the way it always has, with something that looks, behaviorally and emotionally, like mild depression. Now imagine that immune activation is not temporary. Imagine it's not the clean, time-limited response to a vaccine that resolves in 2 days. Imagine it's a chronic, sustained, low-level immune activation, driven by a dysregulated gut microbiome, by a leaky gut lining, by the constant low-level inflammatory signaling we've been discussing. The brain stays in that protective shutdown mode indefinitely, not as a temporary response to a real threat, but as a chronic state, because the signals telling it there is a threat never stop arriving. That is what an increasing body of research suggests is happening in a significant subset of long-term depression cases. It is, in a meaningful sense, the brain getting a constant, unresolvable illness behavior signal from an immune system that is stuck in the on position. And the root cause of that is not in the brain at all. It's in the gut. This has profound implications for treatment. If you address the gut, reduce the systemic inflammation, restore the microbiome diversity, repair the gut lining, you change the signal. The brain stops receiving the illness activation cue. And for some people, that is the intervention that finally moves the needle on depression that has been treatment-resistant for years. I want to be clear. I am not saying stop your antidepressants and eat more yogurt. That is not the message. The message is that the gut-brain connection is mechanistically real. The immune pathway to depression is scientifically credible. And addressing gut health is an evidence-based adjunct to, not a replacement for, appropriate mental health care. I want to share with you one of the most striking pieces of epidemiological evidence I've come across in recent years. And it involves Parkinson's disease. Parkinson's is traditionally understood as a neurological condition. It involves the loss of dopamine-producing neurons in a region of the brain called the substantia nigra. The hallmarks are the motor symptoms, the tremor, the rigidity, the slowed movement, and there are also significant non-motor symptoms, including cognitive impairment, mood changes, and sleep disruption. It's a disease of the brain. That's how we've always categorized it. But here is the finding that has, quietly but significantly, begun to reshape how researchers think about Parkinson's. Approximately 90% of Parkinson's patients, 9 out of 10, report chronic gastrointestinal symptoms like constipation and bloating and abdominal discomfort. And they report these symptoms not at the same time as their motor symptoms or after. They report them 10 years earlier, 10 years before the first neurological tremor, before any motor symptom that would prompt a neurological evaluation. A full decade before the disease arrives in the brain, there are already clear, persistent gut symptoms. The scientific explanation for this centers on a protein called alpha-synuclein. Under normal circumstances, alpha-synuclein performs various functions in neurons. But under conditions of gut dysregulation, an unhealthy microbiome, chronic intestinal inflammation, alpha-synuclein can begin to misfold. And misfolded alpha-synuclein aggregates into what are called Lewy bodies, clumps of protein that are the pathological hallmark of Parkinson's disease. And these Lewy bodies form in the gut first, in the neurons of the enteric nervous system, and then slowly, over the course of years, possibly a decade, they migrate upward along the vagus nerve, moving from the gut toward the brain, progressing through neural tissue until they reach the central nervous system. This is the gut-to-brain pathway of Parkinson's disease. The research behind this comes from some of the most rigorous epidemiological work in the field, including data from populations with vagotomies, surgical severing of the vagus nerve, performed historically for ulcer treatment, which show significantly reduced rates of Parkinson's. If you cut the highway, the protein clumps can't travel. What this represents, from a preventive medicine standpoint, is almost incomprehensible in its implications. A 10-year window, a decade of time, between when the problem begins in the gut and when it manifests in the brain, during which intervention might interrupt the pathway. If we can identify early gut dysregulation, if we can address the microbiome environment in which alpha-synuclein misfolding occurs, if we can reduce the intestinal inflammation that creates the conditions for this to happen, we may have a window to prevent Parkinson's disease before it ever crosses the blood-brain barrier. We don't have a definitive protocol for this yet. The research is still developing. But the mechanistic pathway is credible, the epidemiological data is compelling, and the implication is clear. Gut health is not just about digestion. It is about neuroprotection. It is about brain health on a 10-year timescale. The gut is the earliest warning system we have for some of the most devastating neurological diseases we face. All right, let's talk about what you can actually do. Because everything I've described so far has been pointing to the same fundamental intervention, taking care of your gut microbiome. And the question is, what does that practically mean? The most important insight here, and I want to emphasize this because I think it corrects a significant misconception in how most people approach gut health, is that the goal is diversity, not dosage. Most people who think about gut health think in terms of probiotics. They take a capsule with 5 or 10 bacterial strains. They eat one particular type of yogurt. They focus on getting more of one thing. But the microbiome is an ecosystem. And like any ecosystem, its resilience and its function depend not on having a lot of one species, but on having a rich variety of many species interacting with each other. The Amazon rainforest is not healthy because it has an enormous amount of one tree. It's healthy because of its extraordinary biodiversity. Your microbiome is the same, and different microbial species have different dietary needs. They thrive on different fibers, different polyphenols, different plant compounds. Some love the compounds in coffee. Some love the fibers in legumes. Some love the polyphenols in dark berries. Some love the inulin in chicory or garlic or asparagus. To keep all of them well fed, you need variety. Professor Tim Spector's research group developed what they call the 30 plants a week target as a practical framework for achieving this diversity. The concept is simple: aim to consume 30 different plant foods over the course of a week. Now, when I share this with patients, the first reaction is often, "That sounds impossible. I don't think I even know 30 different plants." But the definition of plant food here is generous and inclusive. Herbs count: a pinch of oregano, a sprig of rosemary, a leaf of fresh basil. Spices count: cumin, turmeric, black pepper. Coffee counts. Dark chocolate counts. Every different variety of fruit, vegetable, nut, seed, legume, and grain counts as a separate plant. A single meal, Pasta with a tomato sauce containing garlic, onion, basil, oregano, a glass of red wine, and a mixed green salad might contain 8 to 10 different plants. The 30-plant target, when you map it against a week of varied eating, is more achievable than it sounds. And the results from Spector's research, tracking participants who implemented the daily 30 approach, are genuinely compelling. 70% reported improved digestion, 43% reported meaningfully higher energy levels, and 45% reported an increase in general happiness. Improved digestion, more energy, more happiness from eating more variety of plants. Not from a pharmaceutical supplement, not from an expensive intervention. From deliberately choosing a different vegetable at the grocery store. From adding a handful of walnuts. From trying a new herb. I also want to share a specific example that I find delightful because it illustrates how targeted the microbiome's needs actually are. There is a bacterial species called Lacanobacter that specifically responds to coffee. When you drink your morning coffee, this particular species essentially has what researchers describe as a party. It proliferates, multiplies, and in doing so produces compounds that actively dampen systemic inflammation. Your coffee is not just giving you caffeine, it's feeding a specific microbial population that is then doing anti-inflammatory work on your behalf. This is what I mean when I describe the gut as a high-end pharmacy. The inventory is stocked by what you feed it, and the pharmacy is open around the clock. Beyond plant diversity, the other major lever for microbiome health is fermented foods. A landmark study from Stanford University found that incorporating fermented foods into the diet reduced markers of blood inflammation by approximately 25%. That is a dramatic, clinically meaningful reduction from a dietary intervention alone. For context, many anti-inflammatory medications achieve effects in a similar range, and fermented foods achieve it without side effects, without prescription, and with a significant number of additional health benefits. The practical framework I use is what Professor Spector calls the 4 Ks: kefir, kombucha, kimchi, and kraut. These 4 fermented foods represent different fermentation traditions, different microbial profiles, and different culinary uses, meaning they can be incorporated across a variety of dietary preferences and palates. Kefir is a fermented dairy drink, or coconut kefir for those avoiding dairy, with an extraordinarily rich microbial profile, often containing 30 or more bacterial and yeast strains. Kombucha is fermented tea, increasingly available in most grocery stores, though quality varies significantly. Look for raw, unpasteurized versions with live cultures. Kimchi is a Korean fermented vegetable preparation, typically spicy fermented cabbage, that is also a source of fiber, vitamins, and a wide array of beneficial bacterial strains. Sauerkraut, fermented cabbage in the European tradition, is one of the most studied fermented foods, with strong evidence for beneficial effects on the gut microbiome. Now I want to address a related trap that catches a lot of well-intentioned people. The health food market has produced a category of products that carry the aesthetic of gut-healthy choices, but deliver very little of the actual benefit. Zero-fat Greek yogurt is the example Spector specifically calls out. The problem is not the yogurt per se. Plain, full-fat, live-culture yogurt is genuinely beneficial. The problem is what manufacturers do to make it zero-fat. The fat, which provides the rich, satisfying texture and carries fat-soluble nutrients, is removed. And to compensate for the loss of texture, starchy fillers and sometimes sugars are added. The natural microbial complexity of a genuine fermented dairy product is often pasteurized away for shelf stability. The product looks healthy. The label says probiotic or live cultures, but the biological reality is often heavily processed food pretending to be something it isn't. There's also a genuinely interesting finding about what Spector calls zombie biotics. fermented products that have been pasteurized, killing the live cultures. You might think these are useless, but they're not entirely without benefit. Even dead bacterial cells, technically called postbiotics, retain their cell wall structures. And those cell wall components, when they reach the small intestine, interact with immune cells there and send a signal, essentially a calming regulatory signal, that tells the immune system to stand down. It's a muted version of the full probiotic effect, but it's not nothing. And finally, a word about calories, because I think the current cultural obsession with calorie counting is actively misleading people about what matters for metabolic and gut health. The structure of food matters far more than its caloric value. Ultra-processed foods, and white bread is the classic example, are pre-digested by industrial processing. The fiber is removed, the complex structure is broken down. The food requires almost no digestive effort. It hits your bloodstream rapidly. It bypasses the satiety signals in your gut. It starves your beneficial bacteria. And it leads to overeating. Some research suggests by an average of 25% more calories consumed compared to equivalent whole food alternatives. The person counting calories and eating a low-calorie diet of ultra-processed food may be consuming fewer calories than someone eating a varied whole food diet, and still be significantly worse off metabolically, microbiologically, and neurologically, because the numbers on the package are not telling the full biological story. Feed the pharmacy. The inventory determines the medicine your gut dispenses to your brain.
Don't forget to hit that subscribe button and join the journey.
Let me leave you with the idea that I think ties everything in today's episode together. Biological humility. The more deeply we look at how the human body actually works, the more we map the communication between the gut and the brain, the more we understand the role of the microbiome, the more we appreciate the evolutionary history of our enteric nervous system, the harder it becomes to maintain the old story. The story where the brain is the king, the body is the servant, and the gut is a digestive tube. We are, it turns out, significantly more microbial than we are human. The cells in your body, approximately 37 trillion. The microbial cells in and on your body, approximately 39 trillion. You are, in terms of sheer cell count, a minority in your own body. They produce compounds that regulate your immune system, manufacture neurotransmitters that influence your mood, send signals through your vagus nerve that shape how your brain interprets the world, and protect or endanger your neurological health over decades. We've been thinking of the brain as a distant monarch, isolated, sovereign, running the show from behind its barrier. But the real picture is of a highly sensitive listener, deeply dependent on the quality of the dispatches it receives from below. Your internal pharmacy is open right now, 24 hours a day. What it stocks, what it produces, what it dispenses to your brain, that depends almost entirely on what you feed it. 30 plants a week. The 4Ks, flossing, whole fermented foods, fiber. These are not complicated interventions. They are, in the most literal biological sense, the way you communicate with your gut, the way you influence what your gut communicates to your brain. The conversation is already happening. The question is whether you're contributing to it. Take care of yourselves, and of the trillions of microbes that are, right now, taking care of you.